There's some folks working on metabolic markers of dietary intake. So are there small molecules in the bloodstream that you can measure or in urine that give you some information about dietary intake? So that's another piece, but there hadn't really been like a data-driven, either DNA or metabolomics way to get high resolution information about the diet until fairly recently. So
I've
maybe
done a food diary, and you're right. If you don't eat something, you're like, or you eat something you shouldn't be eating, you're like, oh man, all right, I'll put it down. And it was very revealing. I had a few friends do it too, and I tend to eat the same thing a lot. Some meals are always the same, some vary. There were some days where I ate 5, 6 times a day, little bits, and then some days I only ate like once or twice. And it was just interesting to see it all. And yeah, taking pictures, like what I saw was when you first start logging, it is a lot of work. But if you have the same meal at like Joe's Cafe every day, once you've got a picture of it and once you log the nutrients and what's in it, then you're like, oh, I just ate at Joe's Cafe that had my normal like fish lunch or something. And then it gets much faster and easier to put in the data. I know it's just anecdotally, I'm just letting you know what I've run into, which may be interesting to you.
Oh yeah, that is interesting. I think that makes a lot of sense. I tried it for a while with image tracking. There was a app sort of collaborator in Europe was developing to try to track your diet through images. And I was doing that, snapping pictures of everything I was eating every day. And it ended up being a lot of work. I think I have a little more of a heterogeneous diet than you, perhaps. I kind of eating different things all the time. So every time I had a handful of peanuts, I'd have to take a picture of them, so it became sort of, annoying and difficult. And I only could keep it up for about 2 weeks. So yeah, it's tough.
Yeah, and one more thing I saw, I forget who it was, but there was the guy that sampled his oral microbiome every day for a year, and it was very stable, except when he traveled overseas and when he got really sick one time. But other than that, it was remarkably resilient and stable to where it was, for the rest of the year. So I'll see if I could find that, but
I know the data set you're talking about because that's my colleague, Lawrence David, who comes into this field. So when he was a graduate student, he collected that data set, and he also had dietary tracking over that same period. And yeah, it was surprising how stable his nutrient intake was over time. Tended to eat a similar set of nutrients from day to day. I don't know how that breaks down to the individual food items he was eating, but on the nutrient level, he had a pretty similar diet from day to day. And so, so now I'll get maybe into the DNA detection of food and where this idea comes from. This is actually an idea that originated in people who study wild animals. So if you wanted to know what a grizzly bear is eating or a humpback whale, you can't ask them. So you have to find some other data-driven methodology. And scientists have turned to metabarcoding. It's a method called metabarcoding where you're amplifying specific marker genes from, dietary sources. And in particular, they're looking at mitochondrial and chloroplast marker genes. And these are little organelles inside of eukaryotic cells. And they contain DNA. And you can use that DNA to tell you which species of animal or plant is in a sample. And so wildlife biologists were using these metabarcoding methods to track diet of wild animals. And Lawrence David, who we just talked about, he pioneered using these same methods for diet tracking in humans. So his lab has been developing, methodologies for extracting DNA and having primers and amplifying out specific portions of genes from mitochondria and chloroplasts to try and, extract out human dietary information from stool, and has a few papers in this area. So we knew all this work in our lab, but there was this lingering idea that maybe you could just randomly sequence all of the DNA coming from poop, right? Not trying to amplify out a specific gene, but taking a shotgun approach, just getting all the DNA and sifting through it, and then just being able to extract dietary DNA from that kind of needle in a haystack problem. It seemed like a maybe impossible task, but Christian Diener, who was a really talented computational biologist in my lab, who's now running his own lab at the Medical University of Graz in Austria, he tackled this problem. It took about 5 years to figure out how to get it to work, but we ended up with a method that we can apply to what's called shotgun metagenomic sequencing. So here we haven't done any PCR. We've just sequenced random fragments of DNA from stool, and it just so happens that there are hundreds of thousands of already sequenced stool metagenomes out there that you can download from public databases, and this suddenly would open up a huge universe of samples to be able to apply our method and extract dietary information that we can pair with the microbiome.